Hi guys, welcome back. I'm Mr. Khor, and today we'll be learning Form 4 Chapter 5, Metabolism and Enzymes. Okay, straight to the point. Today we have a new student. Today I have a new student. Where did he go? Yeah. If you watch, what you call it? Hey boy, no? Okay, later boy. If you watch my earlier video on nucleic acid, you might have seen Patch. He is here today as well. This is previous class new student. Okay, boy, go. This is today's class new student. Okay. Anyway, straight to the point. So, today we'll be looking at Chapter 5, Metabolism and Enzyme. Now, when we talk about the old and new syllabus, okay, sukatan lama, sukatan baru, this used to be part 5 we call it Metabolism and Enzyme. This is actually something new. But you'll be surprised by how short it actually is. Whereas KBSM, there was no such thing as metabolism. Chapter 4, chemical composition of cells, is where you would learn what we have learned in the earlier class: carbohydrate, protein, lipid, and also enzyme. Okay, this is the old syllabus. So old syllabus enzyme is in chapter 4, but new syllabus is called chapter 5, and they added metabolism, but very little part only, very small part. Okay, mari kita mula. Okay, pelajar sekalian. Okay, metabolism, there are basically two types. But before that, straight to the point. Okay, catabolism is whereby large molecules are broken down. Large molecules are broken down to small molecules. For instance, protein broken down to become what? Amino acids. In the previous topic, okay, go and find Patch. Yeah. Think you want to play. Okay, so protein will be hydrolyzed to become amino acid, or it undergoes hydrolysis to form amino acid. This is what we learned in the previous topic. So, catabolism is a breakdown process. And anabolism is the total opposite, whereby small molecules will join together to form a large molecule. We call this anabolism. Okay. Example, the opposite lah. Many amino acids join together in order to form a protein. Yeah. And that's all for metabolism. That's it. Okay, the next part would be enzymes. So enzymes, there'll be two parts. This video would cover part 1, and hopefully just with the next video I'm able to cover the whole thing already. Which means dalam 2 video kita dapat selesaikan satu topik. Okay? Okay, mari kita mula. Now, enzymes here, if you have done in school, guys, can you give me one word? You have done in school, I'm very sure you have learned this. Your teacher confirm would have told you. Can you give me one word for enzyme? One word for enzyme. What is the word? The word is protein. Semua enzim ialah protein. All the enzymes are made up of protein. Enzymes are proteins that catalyze, memungkinkan. Before that, Yeah, enzymes, they are protein. Guys, if I were to ask you, what is the monomer of enzyme? What is the monomer of enzyme? Your answer should be amino acid. Why amino acid? Because the monomer for protein is also the monomer of, the monomer of protein is also the monomer of enzyme. Why? Because this fella is a protein. This fella is a protein. Now, there are proteins that catalyze. Dalam BM kita sebut memangkin, okay? Catalyze, meaning speed up reactions, make it quicker, make it faster. So, guys, again, can you give me one word for enzyme? The word is protein. What do they do? Oh, they catalyze metabolic reactions. So, when your syllabus asks you the necessity, apakah keperluan? Apakah keperluan enzim? Just use the next part. Oh, necessity of enzyme is to catalyze, meaning speed up metabolic reactions. Okay? Now, third part, nomenclature means naming. Penamaan. Nomenclature means naming. So, if you think about enzymes which you know, can you come up with a few for me? Example, lipase. Example, pepsin. Example, amylase. Okay? So, in short, there are two groups of enzymes when it comes to naming. There are two groups of enzymes. One with ASE at the back, one without. Okay, two groups. So guys, naming of enzymes, the first group, kumpulan pertama is whereby the first part would be the substrate name followed by ASE. Okay, let's run through very quickly. If let's say, katakan, let's say substrate name is sucrose. What is the enzyme? Sucrase. What if maltose? Maltase. Lactose, lactase, lipid, lipase. Yeah, this is how you get the word lipase. Urea, urase, and protein, protease. So, ini kumpulan pertama di mana nama enzim akan bergabung dengan nama substrate. It combines with the substrate name, okay, by using ASE at the back. ASE at the back. Kumpulan kedua tak perlu ASE langsung. There's no such thing as ASE. They stand alone. Example, pepsin, rennin, trypsin, as well as erepsin. Alright, if you have my English version notes, whatever I taught you, everything is in. I'm a non-believer. When it comes to copying hell lot of notes. It doesn't work that way. Studying doesn't need you to copy a lot actually. If copying works, you don't even need to attend. You don't have to be here right now. You can just buy a reference book. Just copy everything to a A4 paper, then you should score 100 marks already. Okay, anyway, guys, if you have my English version notes, just to stress again, because many of you who are watching are my existing students. Again, to score bio, I have to keep stressing on this a lot. To score bio, number one, you refer to my notes, attend all my classes, okay? And number two, have one reference book. Should be enough, should be sufficient to get at least A- already. At least A-. My notes are rather complete. If you can cover everything confirm, at least A- is there. Confirm. Okay? Alright, now look at page 16 page 16 on the left hand side, 5.3. Even before we start, please use a red pen, draw 3 stars. Draw 3 stars for this area. There are quite a lot of characteristics of enzymes which you have to know. Now, first of all, the one which all of you already know, all enzymes are protein. Okay, full stop, that's all. All enzymes are protein. Yeah, okay, if you see this in my notes, please draw 2 stars because this is quite important. Enzyme reaction is specific. What it means is, one enzyme only catalyzes one type of reaction. Okay? Sejenis enzim hanya memangkin sejenis tindak balas. For instance, revision, or before that, check first. Maltase catalyzes the hydrolysis of maltose. Revision, what we learned in chapter 4, carbohydrate. When we break down maltose, what do we get? What are the 2 monosaccharides that we get? Apakah kedua-dua monosaccharida yang kita akan dapat? Jawapan, glukosa. Okay, answer will be glukosa. Now, in the presence of water, maltose will be broken down to glucose. But in the presence of maltase, this will speed up, mempercepat, speed up the reaction. Means we can have more. Later we will learn, all these are called products. All these are called products. We'll get more products, or we'll get the same amount of products in a shorter period of time. Okay, the time taken will be shorter. Now, example 2, sucrase. Okay, quickly tell me, what does it break down? This one is sucrose. Sucrase breaks down sucrose in order to form glucose and fructose. Okay. So, guys, enzyme reaction is specific means one enzyme only act on one type of substrate. Yeah. All these are called active site. Okay, all these are called active site. Tapa active. Active site is where the substrate, we call this a substrate. Earlier I mentioned all these are products. So before products, they are considered substrate. Substrate will enter the maltase. Maltose will enter the active site. Reaction takes place and we get products. These 2 are products. And if you realize, okay, check. So before the maltose enter, after products are formed, there are no changes to the enzyme. So we say that enzymes are not changed or destroyed after reaction. Yeah, this is one of the characteristics, but not overly important. The size of enzyme is always bigger than the substrate. Okay, now this is the important one. Can you please draw 3 stars? Can you please draw 3 stars for this characteristic, sensitive to temperature? Now, let me ask you, what is your body temperature, guys? What is your body temperature? Some of you 36.9, some of you might be thinking 37, I don't know, maybe some of you thinking 28, 16, that's up to you lah. But okay, the reason why your body temperature is as such is because at this particular temperature, enzymes work at its best. Enzymes work at its best. You can see here, in terms of rate of reaction, this is the highest point. Because at this temperature, rate of reaction is at the highest point. We call this, guys, the optimum temperature. Suhu optimum. Suhu optimum. Now, if you want to know just some basic stuff, this is okay lah. This is okay. If you are aiming to score, you have to know much more. Okay, let me explain in greater detail. Have a look. So on my y-axis would be the rate of reaction, x-axis temperature. Okay, assumingly this is 10 degrees Celsius, 20, 30, 40, okay, 50, 60, and so on. Okay, now this is our graph just now. Okay, hold on. Okay, this is our graph. Alright. Okay, a few things to know. Okay, number one, at low temperatures, we say that rate of reaction is low. At low temperatures, okay, rate of reaction is low. Why? Because, okay, the reason is because at low temperatures, enzymes are not active. They don't work that efficiently. Okay. Now, second point. When the temperature increases by 10 degrees Celsius, rate of reaction will double. Okay. Apabila suhu meningkat sebanyak 10 suhu darjat, okay, kadar tindak balas akan berganda ataupun ganda dua, double. So, guys, let's say this is rate of reaction A at 10 degrees Celsius. When I increase 10 degrees Celsius, it is now 20. Can you tell me what the reaction is here? For every 10 degrees Celsius increase, rate of reaction double. So initially A, now is 2A. Now it's 2A. And what if another 10? This would be 4A. Okay, this will be 4A. So what if I draw this one? Now, there's one catch. This only applies below 40 degrees Celsius. Yeah, this only applies below 40 degrees Celsius. Or some books will put 37 degrees Celsius, which is fine. Now, one thing you all have to know is when it comes to what we call the optimum temperature, some books will put 37, some books will put 40. Either one is fine, because SPM also they do it that way. Sometimes they put 37, sometimes 40. So now this is actually the third point. Okay, my graph here shows very clearly 37 degrees Celsius would achieve the highest point. Let's say rate of reaction X, which is actually the highest. Okay, highest rate of reaction. Now, due to that, we call 37 the optimum temperature. Optimum temperature. Now, at optimum temperature, we say most products are formed. Yeah. Okay. Okay. One last thing. Color is nice. Red, blue. Okay, one last thing. Now, in exam, they always like to ask you what happens if the temperature is 60 degrees Celsius and above. So this leads us to point 4. Now, guys, temperature higher than 60 degrees Celsius, there will be any reaction here. Ada tindak balas tak? Tak ada langsung. There will be no reaction. There will be no reaction at all. Meaning no products. And if exam asks you why, this is the key point. We say enzymes are denatured. Denatured. Enzymes are denatured. Dalam BM, dinyah aslikan. Telah dinyah aslikan. Already denatured. Now, to explain denatured, okay, let me explain here on the left hand side. At optimum temperature, this is how our active site looks like. But when it is, let's say I take 70, let's say I take 70 degrees Celsius, this would be the active site of my enzyme now. Hello students, what have happened? Okay, denatured means the active site change shape. The active site change shape, substrate cannot bind, therefore no reaction and no products. Okay, all right. So if you are fine with it, you can straight away proceed to my notes. Everything is on page 19. Yeah, page 19 on the left hand side, point 3. Okay. So if you are aiming to score at least A minus, you must know all these 4 main points. All these 4 main points. Number 1, low temperature, rate of reaction low. Enzymes not active. Enzymes not active. As temperature increase, every 10 degrees Celsius increase, rate of reaction double. And we will hit the optimum temperature, okay, whereby it forms the most product. Why? Because of highest or maximum rate of reaction. Finally, temperatures higher than 60 degrees Celsius, enzymes are denatured. Dinyah aslikan. Kenapa? Sebab tapak aktif telah bertukar bentuk. Okay. Okay, one last one here. Enzymes are sensitive to pH. Enzymes are sensitive to pH. There are basically a few enzymes that you have to know, but let me cover the main ones first. Okay guys, if you have heard of pepsin, where do we find pepsin? In the human stomach, correct? In the human stomach. Is your stomach acidic or alkaline? Of course, your stomach is acidic. Have you wondered why? Kenapa? Why your stomach is so acidic? Your stomach is nearly as acidic as the hydrochloric acid. Okay? It's about pH 1 to 3. It's about pH 1 to 3. Okay, because at this pH, pepsin works at its best. Therefore, the optimum pH for pepsin is pH 2. The optimum pH for pepsin is actually pH 2. Oh yeah, is your mouth acidic or alkaline? Okay, is your mouth acidic or alkaline? If your answer is either one, something must be wrong with you. Your mouth is actually neutral. Okay, neutral. Why? Same reason. Because pH 7 is the optimum pH for amylase. Okay. Now, one more main enzyme that you have to know is called trypsin. Trypsin is found in the small intestine, which we call ileum. Small intestine, which we call ileum, usus kecil. And our small intestine has a pH of about pH 8 to 9. Means a little bit alkaline. Because this is the best pH. This is the best pH for, 8.5 is the optimum pH for trypsin. Yeah. So to remember this well, optimum pH 8.5. You may use the first alphabet of each enzyme, P, A, T. Remember PAT, PAT. Okay, but don't say it twice, Yeah, you know why, don't say it twice, Okay. Now. If you have my notes, I actually included, or if you have my notes, you need to include this. And my notes have one extra one running here. So just take note of these 4 enzymes. 1, 2, 3, and then 4. Okay. So the characteristics are all on page 16 in my notes. The English version. Okay, yeah. We conclude the first part of enzymes here. All right. So if you are keen, you can log on to the next video, which explains hopefully everything else of what you need to know for enzymes. Okay, guys. Cheers.